Hey class, let's pick up with the different kinds of cells. So we're still in chapter 1 of our text. Let's talk about cellular organization. The two big categories used to be eukaryote and prokaryote. Now there's three, sort of. If you look at our slide, we still have it in two big bullet points, but the second bullet point's broken up into two categories. So eukaryotes, this is the one that we know and love very well. They have lots of organelles. So if we have a true nucleus or true kernel life form, it has organelles. Man, I bolded that. It must be important. It has lots of membrane-bound structures. These bacteria and archaea do have ribosomes in them, which are technically an organelle, but ribosomes are not membrane-bound. Examples of membrane-bound organelles include the nucleus, mitochondria, chloroplast, Golgi apparatus, the rough ER, the smooth ER, lysosomes, peroxisomes. I could go on and on. So we have lots of membrane-bound organelles. Some are going to be microorganisms, some are going to be macroscopic. When we think microorganisms, think small. When we think macroscopic, think big enough to see with your naked eye. Bacteria and archaea are a lot smaller than a eukaryote. They are approximately 10 times smaller. Yes, there are exceptions, but generally speaking, bacteria and archaea are really small compared to a eukaryotic organism. Bacteria and archaea lack organelles, with the exception of ribosomes, and every single bacteria and archaea is a microorganism. There are no multicellular bacteria and archaea that we can see with the naked eye. Viruses. Viruses are kind of a weird category. They don't live independently by themselves. They must have a host cell to infect to reproduce. So they are somewhere between a molecule and a cell. And I love the way this is phrased right here. They're somewhere in complexity. There exists a level of complexity between a large molecule and a cell. A molecule all by itself is by definition not alive. A cell by itself is by definition the smallest unit of life. And then somewhere in between is the virus, making our definitions poor and ineffectual. All viruses are going to have a little bit of genetic information. These are going to be in the form of DNA and RNA, and these are going to be surrounded by a protein coat, and every once in a while, a lipid-based membrane. Outside of a host cell, our viruses are unable to reproduce, by definition. When they get into the host cell, though, their genetic information is released into the cytosol of the host cell and then reprograms the host cell from the inside out to do its bidding. You could think of a virus as something that makes a zombie or a slave out of the host cell. These viruses are incredibly small. Typically, you can only see a virus with an electron microscope. There's only one exception, one virus that I can think of that's 1.2 microns across and that you can actually see with a light microscope. But generally speaking, viruses are so small you cannot see them with regular microscopes in the laboratory. So, let's do a concept check. This concept check is actually going to be related back to earlier PowerPoints in this chapter. What is the organism? What's the largest and then the smallest organism? So if we think back to earlier in this presentation, we had mentioned how helminths are always multicellular organisms. Helminths are the big guys on the block. So we start with D, helminth as the largest. Then we have protozoas. Protozoas are a eukaryotic organism that can be single cellular. So they are going to be the second largest. And then we have viruses and bacterium. Between a virus and a bacterium, hopefully at this point, based off the slides I just talked about, you're able to say that, yes, a bacterium is bigger than a virus. I'll repeat that because it's important. Bacteria are bigger than viruses. So, to list these in orders from largest to smallest, we had helminth, then protozoa, then bacteria, and then virus. Throughout history, there have been a lot of theories to talk about how microbes came to be. Spontaneous generation used to be a very popular one. It's believed that there used to be some invisible life force, kind of like the ether in outer space, which has since been disproven. As a side note, when someone says that something has disappeared into the ether, they're referring to something disappearing into the nebulous force that holds the solar system together. That has since been disproven. Spontaneous generation was based on the belief that there's some invisible life force, kind of like the Force or Gaia, that brought rise to life on our planet. This has since been disproven. The discovery of microbes, the belief of abiogenesis, a meaning without life creation, or the creation of life from non-living sources, was embraced by some scientists. Other scientists advocated for biogenesis, the belief that all life had to come from other life. And this is the one that's currently embraced and taught and has since been supported by the vast majority of evidence. Abiogenesis has been definitively proven to be false for the vast majority of microbes. Biogenesis is the one that holds up repeatedly in experiment after experiment. To get a worm, you need to have a parent worm, so on and so forth. We need to highlight Louis Pasteur. He is a giant in the field of microbiology. He was born in 1822, died in 1895. I included a mugshot of him because he is that important. What did Louis Pasteur do? Louis Pasteur studied microorganisms related to the fermentation of beer and wine. He also invented the process of pasteurization, which we commonly do today for our fluids, to remove microbes from our fluids to keep the fluid shelf safe. It's kind of a tongue twister there. One of the things that Louis Pasteur did in his experiments is he came up with the idea of making a swan-necked flask. These swan-necked flask experiments were used to disprove abiogenesis, the creation of life from a non-living thing. He would take this swan-necked flask and fill it with broth. And then and when I say broth, I mean like beef broth extract or the liquid from a soup. And then he would take that liquid and he'd heat the living daylights out of it. And he would boil it for hours and hours at a time in a water bath. After he was done boiling the heck out of the broth, if he exposed that broth to dust from the air, there was microbial growth because there's lots of bacterial and mold spores floating around in the air. But the flasks that were exposed to the air but not to dust showed no microbial growth. So how did he do this? How could you expose a flask to air and dust and one to air and no dust? In these situations, he would seal the flask so that there was air inside of the flask to expose it to air and not dust. Or he could take the swan neck flask and have the opening come up and then point back down again. Let's look at a picture. So here we have a swan neck flask. This swan neck flask can be exposed to the air. It's still possible for the air to get in and out of the swan neck flask, but any dust that would float through the air would settle right here in this bottom portion, and the dust would not float up to the surface to get down into the broth. If Louis Pasteur broke the top of the flask, dust would be able to get down in there and cause growth, but if he left the swan neck flask in place, the dust would accumulate in this divot, or the S-trap, in the swan neck flask. And it was found that when dust could get in, there would be microbial growth. When no dust could get in, there'd be no microbial growth. Ergo, there must be something in the dust causing the growth of microbes. Another giant in the field of microscopy is Robert Hooke. He was born in 1635 and died in 1703. He studied household objects and plants and trees using homemade microscopes. He was one of the very first individuals to draw what he saw underneath a microscope and publish it, which is why he gets a lot of credit for what he did. Was he the very first one? No, he was not the very first guy to actually do this, but he was the very first guy to widely publish his findings, so he gets a lot of the credit for doing it. Robert Hooke, because he was the first to widely publish his findings, is generally given credit for inventing the microscope, even though the microscope did exist before Robert Hooke. If we look at the top of the screen here, we can see an actual drawing that Robert Hooke made of the cork cells that he was looking at underneath the microscope. And we can take this actual drawing, we can compare it to a photomicrograph, or a photograph of cork cells. So if we look at the drawing and compare it to the photograph, we can see that Robert Hooke had madly awesome artistic abilities. He was one heck of an artist. He could draw a very accurate representation of what he saw underneath the microscope. Another giant in the field of microscopy is Antoine Van Leeuwenhoek, and I'm very positive I pronounced that wrong. It's a French name, so of course I said it wrong. He was born in 1632, died in 1723, and he made microscopes to study fabrics. His background, his training was in textiles. He was a business owner that sold fabric for a living. And because he wanted to better understand the fabric he was selling and make better fabric, he started making microscopes. And then, by gosh, by golly, he decided to look at a drop of water on one of his microscopes. And he found that there were things moving around inside of the water. And he called those animalcules. He found animalcules in drops of rainwater. He found them in pond water. He found animalcules in the scum that he scraped from his teeth. Antoine Van Leeuwenhoek made many, many microscopes over the course of his lifetime. He's created over 250 microscopes. To do this process, he learned how to blow glass. He learned tinsmithing. He learned goldsmithing. He learned how to process the raw materials, the glass, the metal, to manufacture everything from scratch to make his microscopes. This guy was a stud when it came to manufacturing microscopes. And he became very, very good at what he did. And he made the best microscopes in his day. He was capable of magnifying objects to 300 times total magnification. And because of his work with microscopes, he's given credit for making the first precision microscopes. Robert Hooke was making microscopes before Antoine Van Leeuwenhoek, but Antoine Van Leeuwenhoek was making the best microscopes of his time. So he's given credit, and I use quotes because it's kind of nebulous. There's not a very good historical record present, but he's given credit for making precision microscopes. There are some other big players in the history of microbiology. I did not include their profile pictures because, first of all, it's hard to find, and secondly, their contributions were not quite as monumental. Still very important to make the textbook, though. We have Oliver Wendell Holmes and Ignace Samoa. These two gentlemen together describe the importance of hand washing in preventing disease in a hospital setting. They ask such questions as, where did mom give birth? If mom gave birth in a hospital setting, she was more likely to contract an infection than if mom gave birth at home. And maybe that's because there's some kind of invisible pathogen or invisible disease that's being moved around in the hospitals. They found that if there was more hand washing involved, or if mom wasn't treated by someone who was just with another patient, that she wasn't as likely to get the infection. And they also found that if a physician had just been touching a dead body or performing an autopsy on a corpse, and then would go and help mom give birth and handle mom's open wounds after she gave birth, that mom was very likely to contract a disease. And if the physician would wash their hands in between handling the corpse and handling mom, that she was much less likely to get a disease. This is monumental stuff. We take it for granted now that you wash your hands in between patients so that you don't have contamination from patient to patient, or you minimize contamination from patient to patient. These are the gentlemen that helped standardize that practice, or at least came up with the data, the evidence to show that it was worthwhile. We also have Ferdinand Cohn and Joseph Lister. Ferdinand Cohn is a gentleman who discovered that there are heat-resistant endospores, that even if you boiled a liquid for an extended period of time, these endospores would not be killed off at boiling temperatures, at 100 degrees Celsius. And then there's Joseph Lister. Joseph Lister is the gentleman who pioneered the use of aseptic technique in surgery to minimize the amount of infection. He is also the namesake of Listerine mouthwash, which I find to be kind of a cool bit of trivia. We also have Louis Pasteur, as I had mentioned earlier. He's a giant in the field of microbiology. He invented the process of pasteurization. He is also the very first individual who linked human disease to infection. Up until Louis Pasteur, it was believed that most diseases were caused by ethereal spirits and a lot of pseudoscience, things that weren't very well established by the data, but had anecdotal support. He was clearly able to establish that diseases were caused by infection from microbes. We also have Robert Koch. Robert Koch is famous for Koch's postulates. Koch's postulates were a series of steps, experimental procedures, that could be used to establish whether or not a disease was caused by a specific pathogenic organism. He used his own postulates to show that Bacillus anthracis caused the disease anthrax, way back in 1875. Some other recent advances that we had in the field of microbiology involved the discovery of restriction enzymes way back in the 1970s. Restriction enzymes are enzymes that cut DNA at very specific spots of repeating units that are at the same forwards and backwards. If you can read it the same forward and backwards, it's referred to as a palindrome. And these restriction endonucleases will cut DNA at a palindrome. By the way, my favorite palindrome is taco cat. It's spelt the same forwards and backwards. Back in the 1980s, biofilms were being discovered. We started to realize that bacteria can form communities, and that when bacteria form communities, they behave differently than bacteria that are floating in a solution all by themselves. Kind of like middle school kids. When middle school kids gather together at the lunch table, they definitely act very differently than middle school kids that are sitting by themselves in their bedroom. I digress. Back in the 2000s, small RNAs, siRNAs, small interfering RNAs, were discovered and were studied. It was found that small pieces of RNA, small siRNA, small interfering RNA, can modify the expression of other genes. Up until the 2000s, it was very strongly believed that we went from just DNA to messenger RNA to proteins. But now we found that there are some RNA molecules that have useful purposes in the cell other than making a protein. And now, since 2010, what we've been focusing on for most of our research has been the identification of the human microbiome. The fact that there are lots of microbes inside of a human being, and that these microbes play a very important role in human health. We'll talk about these more in later chapters. Anecdotally, the categories of probiotics, prebiotics, and fecal matter transplants all stem from the study of the human microbiome. So, subject. Who is the scientist who built the first high-resolution or precision microscopes? Was it Pasteur, Hooke, Leeuwenhoek, or Ignace Samoa? If you don't remember, you can rewind this presentation or flip back in your PowerPoint slides. Hopefully, you were able to come up with right here, Antoine Leeuwenhoek as your answer. So C, Antoine Van Leeuwenhoek. I'm not even going to try to say it. Am I kidding? And that wraps up this section, the brief history of microbiology.